It was a NIH granted fund study. We initially started this project to looking at the tissue specific relaxations, which means that whenever you place any object into the MRI gives signal, especially from the brain. We get the signal from multiple components. One is from cells, cellular structures, obviously, on iron contributions or other molecules, and also the deoxygenated blood. That also contributes a large part of MRI signals. So our lab, or my professor, has been in this field for several years, and he developed a concept called where you can discriminate these components, because normally they combine all these things. And people think that signal relaxation over the time, it's called as mono exponential, and they just fit it and find this T2 star, R2 star. That is what they do. But in what he found in the beginning of this echo timings, he found there is a quadratic behavior that attributes to the blood, like deoxygenated blood, which is called static bold effect. And then later part, it's mostly contributions of the cellular effect. So based on that modeling, we kind of trying to find this R2T star, which is tissue-specific relaxations that tells you the cellular integrity of the brain. In a way that it's a quantitative measurements. We have started this project for distinguishing the cell's integrity in the brain when it comes to mild cognitive impairment or mild AD symptom populations compared to healthy controls. That's what our aim in the beginning. But later on, when we are developing, we realize that R2T star is not just cellular structure, it's specific to the neurons. That's part of the neuronal relaxation, because neurons act as an endogenous contrast in the brain. As a result, it provides largely signal to the MRI signal. And then what we understood when we look at the T1 weighted images, there is no contrast differences of the tissue structure. Even people have a mild AD, they have the structure, hippocampus, specifically hippocampus structure. There is no intensity difference within the structure, whatever you get the remaining after the atrophies. And what we found in R2T star, there is large part of the structure is very dark, which is very hypo-intense in the sense in imaging term. And then certain part of, the R2T star normal brain is around 12 to 13 second inverse. But in our case, we saw these hypo-intense is around 1 second. And that is the lower boundary we put. And we say that why we see this kind of hypo-intensity. And then we kind of looked into this relation between this gene profiles atlas of the neuronal structure and our measurements. We drawn an equation, which is a linear equation. When R2T star less than 5.8 second inverse, then the tissue does not contain any neurons. When R2T star greater than 5.8 second inverse, the tissue contains a neuronal cells or certain concentration of neurons. I don't know how much concentration. There is a concentration of neurons that can provide a signal to the MRI or detectable signal for MRI. So that's what we came to an idea. And then these two different tissues now. Well, you get the T1 weighted gives you one tissue, which is a gray matter. But when you look into R2T star, it gives you two type of tissues. One is dark matter. One is we call as a viable tissue, which is a functional tissue where you have certain type of neurons. And it's largely when we looked at the normal populations, we still see this dark matter. We don't know why we see this. And when I did some literature review, they says that there's a pathology existing in normal aging brain, like after 65 years old. But when it comes to the mild AD, this dark matter significantly increased a lot. Like it's a mild AD compared to healthy controls. But what is striking over here, when we call as preclinical AD, where population doesn't have any cognitive impairment symptoms or cognitive decline symptoms, but they have this amyloid depositions in the cortical region, cerebral cortex region. So that population we consider as a preclinical AD, which is considered as a prior to population going towards mild cognitive impairment. So we looked at the significant group differences between normal and preclinical, which is not observed by this standard clinical setting MRI measurements. Now they coming to this ATN framework. Maybe you heard of this ATN framework. A is amyloid and T is tau and N is neurodegeneration. That based on these classifications, they decide which brains is AD, certain AD, which brains are not certain AD. And for neurodegeneration is mostly assessed by morphological MRI measurements. But that is not showing any significant differences between normal and preclinical AD subjects. But our measurements shows there is a significant differences between the normal brains and then the preclinical AD subjects. We did the classification. We found around like 80% of the classified preclinical as a preclinical, which is quite exciting compared to volumetric classification, where it was only 69% classification has been done just with the volumetrics. So I would say our technique is in sense or sensitive to the neurons, but most part of the thing, everybody know that volume shrinks with a mild AD people. Over the time period also, there are two causes. One, the aging, shrinking, another one, pathology shrinking. Of course, when there is a pathology, it goes faster compared to aging, shrinking, or atrophy. But previous literature shows that prior to this shrinking, compared to shrinking, the loss of neurons within the whatever remaining, loss of neurons are very high. And what we found also, that's histopathology studies. They found approximately 50% neuronal loss higher compared to volumetric loss. And we found the similar like 40% higher neuronal loss or dark matter loss compared to the volumetric loss. So our measurements are showing that there is this technique is sensitive for neuronal loss. As a result, we call like dark matter is a biomarker sensitive enough for distinguishing these two. It's a prominent biomarker for detecting or understanding the preclinical Alzheimer cases. Coming to the long-term goals about our study, we wanted to look at the trajectories of the brain over the period, which we haven't done. So far, we have done only approximately 150 subjects, but we're presenting only 70 subjects brain analysis. So far, a lot of them are motion artifact because our measurements is sensitive for motion. We're trying with deep learning algorithms to reduce this motion algorithm, but the most important thing is our sequence time is 6 minutes only. Our sequence provides not only neuronal loss or dark matter and this viable tissue, but also it can provide the whole brain analysis of any etiology that existing with the Alzheimer's, MS, multiple sclerosis. It can detect a tumor. It can provide it can provide quantitative information of that. So within 6 minutes, you get the structural scans, you get the quantitative scans of the entire brains. So that in that sense, we are quite innovative. I recall, I don't see where there are existing techniques in MRI, spectroscopy, or the NODI technique, such as this diffusion tensor imaging, which is another kind of diffusion tensor imaging. That's in neurite diffusion-based techniques, but they have their own things. We have this. Our technique is 1:1:2 resolution, which is quite in good resolution. They're doing good, and we are providing another aspect of information for the brain.